Hello, my name is Sofia Pacedano, and in this video I am presenting our poster entitled Who is not Goniodoridella savigny, proved fall 1933, the challenge of the Indo-Pacific Goniodoridella species complex, Neubranchia goniodoridae. Goniodoridella is a small genus belonging to the family Goniodoridae. The genus was selected from specimens collected in the Gulf of Suez, describing the species Goniodoridella savigny. This species has been reported widespread in the Indo-Pacific Ocean. However, these reports are simply published photographs of the specimens, some of them with brief external description. In 2015, a second species, Goniodoridella borealis, was described from Boismana Bay, Russia. In this work, the authors claim that Goniodoridella savigny could include different cryptic species, and that Goniodoridella borealis was previously confused with Goniodoridella savigny. However, the differences between these species are not well defined, since both of the species lack details of internal anatomy. Furthermore, there are no sequences for molecular studies available for any of them. The authors only indicated that the species were differentiated by color and details of the dorsal processes. However, even the color pattern of Goniodoridella savigny could be questionable, as it is not well described in the original description. Because of this aspect of the existence of a species complex and the wide distribution of the species, we begin to ask ourselves, which of these species are not Goniodoridella savigny? We study specimens identified as Goniodoridella savigny collected from Philippines, Australia, and Papua New Guinea. We obtain partial sequences for the genes CO1, Cystinex, and H3 for 6 specimens and perform Bayesian inference and maximum likelihood phylogenetic analysis. In addition, we calculated and corrected p distances for CO1, Cystinex, and H3, and conducted BPTP and ASAP species delimitation analysis on Cystinex in-group sequences, which is the only gene available for all specimens. We also studied the external morphology of the specimens using photographs of the living animals and laboratory observations, and the internal anatomy of some specimens by dissections. Photographs of the radula were taken under the scanning electron microscope. Specimens here studied cluster in a well-supported clade. However, the molecular results differentiate at least 4 different species. These results are also supported by the species delimitation analysis and the uncorrected p distances. The morphology of 3 of these species were also studied. However, dissections of the species from Papua New Guinea were unsuccessful due to the small size of the animal. When comparing the original description of Goniodoridella savigny with the specimens here studied, we can see that all specimens have smooth rhinophores, 2 papillae in front of the rhinophores and 2 at the end of the body, in agreement with the original description of Goniodoridella savigny. However, the color patterns and the reproductive systems of the specimens cannot be compared due to the lack of a detailed description in the original article. Regarding the radular formula, Goniodoridella savigny was described having 2 lateral teeth on the radula, but the number of rows was not specified, and the morphology of the teeth was not well described due to the small size. Despite this lack of information, Goniodoridella species 1 differs from Goniodoridella savigny having only 1 lateral tooth. Our morphological and molecular results shows that there are several undescribed species of Goniodoridella in the Indo-Pacific Ocean. However, the scant information in the original description of the type species and the similarity between the specimens studied makes Goniodoridella savigny a difficult species to determine. The description of new species of Goniodoridella is very complicated due to the uncertainty of which of the species could be the true Goniodoridella savigny. In summary, A complete study based on morphological and molecular data of specimens collected from the Red Sea is essential to determine the characteristics of the type species and thus be able to elucidate the biodiversity of the genus Goniodoridella. Thank you very much for your attention. If you have any question about the problematic of Goniodoridella, any available material that we can use, or suggestions for improvement or collaboration, please don't hesitate to contact us.